but today we are still online, so it won't be so convenient, but I hope we'll manage to finish our tasks. And the topic of the first lecture this morning is about some of the membranous organelles of the eukaryotic cell. And these are organelles which are usually present in all cell types. Universal cell organelles. Usually some cells are losing these organelles when they become terminally differentiated cells and they do not need them anymore. But usually these organelles are present in every eukaryotic cell. And these are the Golgi apparatus or Golgi complex or Golgi network. These are different names for the same structure in the cell. The so-called endosomes, which are closely related to the function and structure of the Golgi apparatus, and coated vesicles, which are also intimately related to this organelle, but not only. So these are the topics for our first class, and this is the brief schedule for the class today. Let's start with Golgi complex, and let's see what is the structure of this very important membranous organelle, and of course to correlate this structure with function. Golgi apparatus, Golgi network, Golgi complex, Golgi zone, these are all synonymous terms to describe the same organelle, which was described for the first time by the Italian scientists Camillo Golgi. He described the apparato reticulare interno in 1897. Camillo Golgi was studying the structure and functions of the nervous system, specifically the brain. And he introduced a unique staining technique to demonstrate the neurons in the brain, in the nerve tissue. And this staining is famous today, and it is known today as the silver impregnation technique of Golgi, or Golgi stain. And while doing this staining on neurons in the brain or in the peripheral nervous system, you see this staining here in the middle on this quite beautiful picture from the so-called ganglion spinale or dorsal root ganglion. That's a collection of neurons in the peripheral nervous system associated with the spinal nerves, which originate from the spinal cord. And in this dorsal root ganglia, there are very large, beautiful neurons, which are called pseudo-unipolar sensory neurons. And when doing this silver impregnation, he noticed that in the cytoplasm of these neurons, there are many tiny black or dark brown deposits of silver, dispersed usually everywhere in the cytoplasm, but most typically they are seen close to the nucleus of the cell. You see the nucleus here, lightly stained because there are no structures to attract silver there in the nucleus. And he called these depositions of silver or the structures in which silver depositions occur, apparato reticulare interno, or the internal reticular apparatus. Reticulum in Latin means a network, and he believed this is an internal network system inside the cytoplasm. Because of the many discoveries and scientific novelties Camillo Golgi introduced in biology and medicine, he received a Nobel Prize for Physiology or Medicine in 1906. And we will continue to mention Camillo Golgi's name during our lectures and practical classes of histology and anatomy in the future. So it is good to memorize and to remember this eminent scientist of the past. Of course, later, our understanding about the structure of the Golgi apparatus increased enormously, especially with the introduction of new scientific methods of investigation, like fluorescent microscopy. You see here a fluorescent microscopy image, the picture below the silver impregnation. Phase contrast imaging of living cells, and electron microscopy. So here in this fluorescently labeled cell observed under fluorescence microscope, a special substance which emits a light in the visual spectrum, specifically in the green spectrum. This substance accumulates in the Golgi network, in this internal reticular apparatus, and it can be visualized as this interconnected but dispersed vesicles and cisterns around the nucleus of the cell. This is the nucleus of the cell, also labeled with a specific fluorescent dye. This is a living cell observed under the so-called phase contrast microscopy, and this type of microscope is very efficient when observing cells, unstained cells, cells which are maintained in cell cultures, living cells. And here in this cell, which is alive, in the culture media, a special fluorochrome, again fluorescent dye, was a special dye, not fluorescent dye, but a special dye which has orange color was introduced, and this special dye is accumulated in the Golgi apparatus. Again, we see the Golgi apparatus here close to the nucleus. In routinely stained cells, which are stained with hematoxylin eosin, usually Golgi apparatus cannot be demonstrated because the structures of Golgi apparatus stain or have the same staining characteristics as the rest of the cytoplasm, and it stains uniformly pink. But when Golgi apparatus is very abundant, regardless of its features to stain with eosin, it has a little bit lower affinity to eosin. And when Golgi apparatus is very abundant, the areas close to the nucleus, which are occupied by the structures of the Golgi complex, appear lightly stained or almost unstained, and that's how we can see the presence of the Golgi apparatus in such cells. This is a cell which has very abundant Golgi apparatus, also this one and this one. These cells are called plasma cells, and they produce proteins, antibodies. That's why they have many Golgi complex inside. Later, with the introduction of electron microscope, two scientists, Dalton and Felix, described the ultrastructure of Golgi complex in 1953, and they found that Golgi complex consists of a collection of flattened cisterns and associated small vesicles and large vacuoles. This structure is repeated many times in the cytoplasm, and all these cisterns in different elements of the Golgi complex are interconnected together. This basic unit of the Golgi complex is called a dictyosome, and hundreds of such dictyosomes are dispersed in the cytoplasm close to the nucleus or around the nucleus and in close proximity to the endoplasmic reticulum. And each of these dictyosomes consists of these 4 to 6 flattened cisterns. I believe you see them here, and smaller and larger vesicles. The large vesicles are called vacuoles. This structure of the dictyosome was described as resembling the stack of pita bread. So these are the flattened cisterns, 4 to 6. Sometimes in other animal species, the number of these cisterns can reach up to 20, but in the human eukaryotic cells, the dictyosome consists of 7. This is a diagram which shows the same structure of the dictyosome seen under electron microscope. These are the flattened cisterns, smaller vesicles up to 50 nanometers. They are budding from these cisterns, and larger vacuoles which have a diameter of up to 300 nanometers. And this is a scanning electron micrograph of a frozen section, and here we see a mitochondrion, a rough endoplasmic reticulum, and one dictyosome, again the flattened cisterns, the small vesicles which are budding from these cisterns, and larger vacuoles which are also budding from these cisterns. And that's the structure of the Golgi complex and dictyosomes. And here again you see one dictyosome present in the middle and portion of another dictyosome with smaller and larger vesicles, and how these dictyosomes are connected with each other. So they are not separate entities, they are connected with each other. That's why this is called a complex, a reticular apparatus, because all these dictyosomes and flattened cisterns are interconnected with each other. And again, that's a transmission electron micrograph. You see part of the nucleus with its double membrane. These are membranes and cisterns of rough endoplasmic reticulum with ribosomes, these dark granules, and this is one dictyosome with the flattened cisterns, the small vesicles located everywhere, and large vacuoles associated with the dictyosome. The flattened cisterns of the dictyosome can be separated in 5 structural regions. So each dictyosome has 5 structural regions. Each Golgi stack or dictyosome has 5 distinct parts. The first one, which is the convex part, and it faces towards the nucleus and the endoplasmic reticulum, is called cis-Golgi network. Why? Because it consists of a network of interconnected vesicles. Here, the vesicles which are traveling towards the Golgi apparatus start to fuse with each other and form this interconnected cis-Golgi network of such vesicles. Then we have 1 or 2 flattened cisterns which are the cisterns of the Golgi apparatus dictyosome. And then in the human dictyosomes, usually in the middle there is a single medial cisterna, and then again one or two trans cisternae, and then the concave zone of the dictyosome from where these large vacuoles are budding is called trans-Golgi network. So 5 distinct parts, this Golgi network, this is the entry place for the Golgi apparatus, then one or two cisternae, usually a single medial cisterna, then one or two trans cisternae, and trans-Golgi network, which is the exit place from the dictyosome. And proteins and lipids, which are directed towards the Golgi apparatus, enter the cis-Golgi network and then travel from one region to the other region, then to the other region to reach trans-Golgi network. And while being transported through these different functional units of the dictyosome, they undergo extensive modification. We'll see in a few minutes what is the mechanism of this modification, and most importantly, what is the purpose? What are the purposes for these post-translational modifications of proteins and modifications of lipids? And then, when they reach trans-Golgi network, these modified proteins and lipids are being sorted, packaged, and then they bud from this trans-Golgi network, which is the exit place from the Golgi apparatus, in the form of vesicles and vacuoles, which travel to other compartments of the cell or to the cell surface, to the plasma membrane. This unique organization of the dictyosome and Golgi apparatus depends on both the cytoskeleton and most specifically the microtubules of the cytoskeleton and the so-called cytoplasmic Golgi matrix proteins named Golgins. These proteins are long tentacle or forest-like proteins which are attached to the surface of the membranes of the cisterns of the Golgi stack, and they interact with microtubules of the cytoskeleton. That's how dictyosome and flattened cisterns become fixed in the cytoplasm in their defined position. And these forest-like tentacles, the Golgins, also serve as an intermediate which recognizes and guides the transport vesicles traveling towards the Golgi apparatus or leaving the Golgi apparatus. So they serve as structures recognizing and initially binding these transport vesicles to the Golgi apparatus. If we use chemical substances which are disrupting the cytoskeleton, there are such drugs used to treat cancer. Usually, we see a disruption of Golgi complex in these cells, which interferes with the proper metabolism of the cell, and the cell dies. So, interaction between Golgins and microtubules is essential for proper structure and function of Golgi apparatus. And now let's see what happens in this Golgi apparatus, why it exists in the cell. We will return a step before Golgi apparatus and we'll again see what happens in the rough endoplasmic reticulum. You already know that rough endoplasmic reticulum contains ribosomes in which proteins are being synthesized on its surface. And when these proteins are synthesized through a special translocator complex, they enter inside the endoplasmic reticulum. And in this endoplasmic reticulum, with the help of special proteins, a pre-assembled oligosaccharide, which has 3 glucose, 9 mannose, and 2 acetylglucosamine residues, is being attached to the amino group of one amino acid called asparagine. As I told you last week, in biochemistry, according to the coding system of amino acids, asparagine is labeled with letter N. That's why here it is written N-linked glycosylation. This means that oligosaccharide chain is being attached to the amino acid asparagine along the protein. So usually, where there is asparagine present in the polypeptide chain, this preformed oligosaccharide is being added there with a covalent bond. Then, in the endoplasmic reticulum, with the help of such proteins like chaperones, these are guiding proteins, they recognize these glucose residues on the surface of the proteins and guide their folding and the assembly of the secondary and tertiary structures of the proteins. And these chaperones not only guide the proper folding of the proteins, but they also check whether the proteins are correctly structured. If there are some defective proteins which are not able to assume their proper secondary and tertiary structure, the chaperones are guiding these proteins for degradation in the cytoplasm. And additionally, in the rough endoplasmic reticulum, between the amino acid cysteine, disulfide bonds are being formed as part of the formation of the secondary structure of the proteins. And then these newly formed proteins are packed in vesicles and transported to the cis-Golgi network. And you see here endoplasmic reticulum, which sends these proteins and newly synthesized lipids to the cis-Golgi network. And then what happens in the cis-Golgi network and the cis-cisterne of the dictyosome? When these newly synthesized proteins reach the cis-Golgi network via vesicles coated with cautomer protein type 2, they are initially checked whether they need to be sent to Golgi complex or they have reached the Golgi complex by mistake. If there are some proteins which are not destined to reach Golgi complex, these proteins are again packaged in vesicles, but this time coated with cautomer protein type 1 and returned to the endoplasmic reticulum. This process is called sorting, and it happens in the trans-Golgi network zone. Additionally, here in the trans-Golgi network, proteins which will become lysosomal enzymes, we'll discuss lysosomes as organelles during the second lecture today. These lysosomal enzymes are recognized in the trans-Golgi network, and specific mannose residues on these proteins are phosphorylated on the 6th position to become mannose 6-phosphate. This is very important because later you will see here in the trans-Golgi network, these proteins which are marked with mannose 6-phosphate, the future lysosomal enzymes, can be specifically recognized because of the presence of this mannose-6-phosphate and packaged into the primary lysosomes. I will tell you from now that Golgi complex is where primary lysosomes are being formed as organelles. Additionally, here in the cis-Golgi network and predominantly in the cis-cisterne, this mannose-rich residue, which has been added as a preformed one in the endoplasmic reticulum, starts to be modified. Some of these residues are being removed by enzymes and others are added on their place. So this residue starts to be modified. And the enzymes which do this are called glycosidases. These are the enzymes which are cutting and removing monosaccharides from this oligosaccharide residue, and glycosyl transferases. These are enzymes which are adding new monosaccharides to this oligosaccharide chain. All of these enzymes are single-pass transmembrane proteins present in the membranes of the flattened cisterns of the dictyosome. And this is what happens in the cis-cisterne and cis-Golgi network. Now we have reached the middle of the stack. This is the medial cistern. Here, glycosylation continues, and additionally, new polysaccharides the hydroxyl groups of some lipids and also on the hydroxyl groups of serine and threonine amino acids in the protein sequence. So now we have glycosylation on new amino acids, the serine and threonine. This is called O-linked glycosylation because the oligosaccharide chain is added to a hydroxyl group. And of course, these asparagine-linked oligosaccharides continue to be modified further. And then the newly synthesized proteins and lipids reach the trans-Golgi network cisterns and the trans-cisterna of Golgi dictyosome and trans-Golgi network of the dictyosome. Here in these two compartments, something very special happens. Sialic acid, or this is N-acetyl-nevuraminic acid, abbreviated as ANA, N-acetyl-nevuraminic acid or sialic acid, is being added to terminal sugars of some oligosaccharide chains on the surface of proteins and lipids. And additionally, sulfate groups are being added to tyrosine residues of proteins and some sugars. Both sialic acid and the sulfate group is like this. Sialic acid and sulfate groups have a net negative charge, and when you add many such residues on the surface of the protein, you charge this protein negatively, and proteins become negatively charged here in the trans-Golgi network and trans-cisterne of the dictyosome. And specifically in trans-Golgi network, then these already completed proteins are being sorted according to their destination and packaged in transport vesicles. They can become either primary lysosomes, if they contain lysosomal enzymes. They can be directed to plasma membrane, and these proteins will become structural and functional proteins of the plasma membrane, or they can be packed in secretory vesicles, and these proteins are later secreted out of the cell. So That's the structure and function of the Golgi apparatus and its specific subregions, the divisions of the dictyosome. Now, I will briefly go deep into the process of glycosylation or post-translational processing in the Golgi apparatus. You see here the protein, as it is in the endoplasmic reticulum. This is the polypeptide chain, and here on the amino acid asparagine, this preformed oligosaccharide is added and blocked. And then in the Golgi cisterns with glycosidases, some of these sugars are trimmed, and you see what happens with this oligosaccharide. Most of the sugar residues have been removed, and now we have a very short oligosaccharide. But then new sugars can be added. It can be either additional mannose residues that can be added here, 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 and this protein becomes very rich of mannose residues, and it is called a high mannose oligosaccharide linked to the protein. Or other sugars can be added different from mannose. You see here galactose, N-acetylglucosamine, and this sialic acid or N-acetylneuraminic acid with its negative charge. And that's how complex oligosaccharides with negative charge are being added on the surface of the protein. And all this happens in different regions of the Golgi complex dictyosome, in cisternae, in cisternae, or cisternae of the dictyosome. And these are different glycosidases. Golgi mannose 1 is a marker enzyme for the cisterne. It trims mannose residues from this preformed oligosaccharide added in the endoplasmic reticulum. And then we have glycosyl transferases. Here the example is N-acetylglucosamine transferase 1, which binds additionally N-acetylglucosamine. And you see here other such glycosyl transferases which are adding galactose, which are adding N-acetylneuraminic acid or N-acetylglucosamine or other such monosaccharides to form the complex oligosaccharide or mannose-rich oligosaccharide residue. And additionally, together with the glycosylation of asparagine, in the medial cisterna, new glycosylation happens on the hydroxyl group of threonine and serine residues of proteins. And this is called O-glycosylation or glycosylation of the hydroxyl group of threonine and serine. And especially in the cells which are involved in the synthesis and secretion of collagen fibers, this glycosylation happens on proline and lysine amino acids, which are in advance hydroxylated with the help of a special enzyme which uses vitamin C as a cofactor, and this newly added hydroxyl group is being glycosylated by special glycosyl transferases. So this is the glycosylation of proteins in the Golgi complex and endoplasmic reticulum. And here comes the question, what is the purpose of glycosylation? Why we need this to happen to the proteins? Well, there are several reasons, and probably we'll find in the future more with the advancement of the understanding about this process. First, asparagine-linked glycosylation promotes the protein folding in two ways. It has a direct role in making folding intermediates more soluble. So it makes the protein more soluble and aids in its folding by preventing aggregation. And the second point is the establishment of this glycocoat on the surface of the proteins, and these oligosaccharide residues can be recognized by these chaperones, helping proteins, which help the newly synthesized protein to achieve its proper secondary and tertiary structure by aiding in its folding and the formation of disulfide bonds or hydrogen bonds between the amino acids. Second, the presence of oligosaccharides tends to make proteins resistant to enzymatic degradation. Enzymes cannot recognize their specific binding spots on the proteins when the protein is glycosylated, so they cannot degrade the proteins. And that's one of the roles of glycosylation, protection of the newly synthesized protein. Glycosylation can also have important regulatory roles. These oligosaccharide chains on the surface of the proteins can be recognized by specific receptors, and then a specific response can be triggered in the cells by recognizing these glucose residues. And last but not least, recognition of sugar chains by selectins. We discussed selectins last week. This is a type of calci-dependent cell adhesion molecule on the surface of the cell. Selectins bind sugars, and that's how cells become attached to structures of the extracellular matrix or to the surface of other cells, and this aids cell-cell recognition, communication, and adherence. So these are some of the purposes of glycosylation. Why glycosylation is so important, and why we have two organelles dealing with this process: the endoplasmic reticulum and the Golgi apparatus. So that's the structure of the Golgi apparatus. Do you have any questions about the Golgi apparatus and its functions? Okay, let's continue now with the so-called endosomes. Let me explain briefly what you see here. Okay, there is a question about O-linked glycosylation. Okay, let's move to this slide here. So, N-linked is to the amino group of the amino acid asparagine, and glucose residues are added here to this amino group. O-linked glycosylation happens on a hydroxyl group of some of the amino acids, either serine, threonine, or in the proteins which will become collagens, hydroxyproline and hydroxylysine. Again, hydroxylated with hydroxyl group. And the sugar is added to this hydroxyl group. Hence, O-linked and N-linked glycosylation. So for the exam, it the amount of information you present about each topic you have at the final exam defines the mark. And you know, in Bulgaria, the marking system which you use the information you provide for each structure, for each organelle, for each tissue, the higher your grade will be. So you decide how much you wish to know about each organelle, about each type of tissue. So that's a very complicated question. Do you have to learn all the information from the slides? No, that's a university. It's not an elementary or high school. We are not forcing anyone to learn or to know everything. You decide what is important, but then there is a grading system. So the more you know and the more you present during the exam, the higher your final grade will be. So if this answer helps, that's what I can tell you about how much you must know for the exam. Yes, lab manuals are checked during the exam. So not for beauty of drawing or how perfect you have described the structures, just as a verification that you were present during your practical classes and you have worked there, you have studied efficiently while being present in the lab. So that's why we insist you to present your lab manuals during the final exam. Yeah. Let's continue with endosomes. Endosomes are membrane-bound compartments. Think of this as spaces surrounded by membrane inside the cytoplasm in the form of smaller and larger vesicles. And usually these endosomes are located in the region between Golgi complex and plasma membrane. So this diagram here shows the nuclear envelope, and here is the nuclear content. Then the endoplasmic reticulum, one dictyosome of Golgi apparatus, the endosomes, and the plasma membrane, and here is the extracellular space. And endosomes are an intermediate between Golgi and extracellular space and the inside of the cell. And endosomes are the first to receive these proteins, lipids, and components which the cell produces or which the cell takes from outside. And we have early and late endosomes. Early endosomes are the initial structure which has been formed, and then it matures and changes into a late endosome. So endosomes are quite dynamic compartment in the cell. It depends a lot of the functional activity of the cell. The more active the cell is, the more endosome, a well-developed endosome compartment it has. And think of it as an intermediate between Golgi and plasma membrane. And as you saw, all these transportations in the cell from endoplasmic reticulum to Golgi complex, from Golgi complex to the endoplasmic reticulum, between the sisters of the Golgi complex dictyosome, from Golgi complex to the plasma membrane or to other organelles in the cell, all these transportation processes happen via vesicles. And from all these organelles, vesicles are budding. And until now, what we know about the vesicles is that there are at least 3 different types of vesicles present in the cell. Why 3 different types? Because initially, when the vesicles are being formed, so if this is the membrane of the endoplasmic reticular cister, when the vesicle is formed, it buds from this cister. And on the next stage, it separates in the form of a free vesicle from this cister. In the process of the formation, the vesicle covers with a special protein coat, which is firmly attached to the surface of this vesicle and marks the vesicle. This protein coat establishes the type of vesicle, and based on the proteins which are located on the surface of these vesicles, we have 3 types of coated vesicles. The most well-known and the most abundant type are the clathrin-coated vesicles. Then we have these vesicles coated with cautomer proteins, cautomer type 1 protein and cautomer type 2 protein. And here you see electron micrographs showing different vesicles, clathrin-coated, cautomer type 1 coated vesicles, clathrin-coated vesicles. Each type of vesicle is used for different transport steps. We have seen that cautomer type 2 coated vesicles are used for transportation from endoplasmic reticulum to cis-Golgi network and cis-Golgi sisters. Then the retrograde transport from Golgi network to the endoplasmic reticulum is via cautomer type 1 coated proteins. Bidirectional transportation between sisters, anterograde and retrograde of Golgi complex dictyosome happens again with cautomer type 1 coated vesicles. And then from trans-Golgi network, clathrin coated vesicles are budding. Also, the plasma membrane during the processes of endocytosis also produces clathrin coated vesicles. And you see here the endosomal compartment between the Golgi complex and plasma membrane. So the clathrin-coated vesicles are the most well-studied vesicles in the cell. These vesicles were only discovered recently, at the end of the last century, in 1980s, via electron microscopy. So we still don't know much about the structure and how these vesicles function, but yet we know enough about them, and specifically about the clathrin-coated vesicles. So the major protein component of clathrin-coated vesicles is the protein clathrin itself, which forms an outer layer or coat around the vesicle. Each clathrin subunit consists of 3 large proteins marked with red color here and 3 small polypeptide chains. And this special structure is called a troiscillion. And many such troiscillions, this is an atomic force microscopy, many such troiscillions are located on the surface of the vesicle and tightly attached to the surface of the membrane of this vesicle, and that's a scanning electron micrograph which shows this three-dimensional lattice of troiscillions around the coated vesicle. And this is a high-power transmission electron micrograph showing just one of these troiscillions, how they really look. This is a diagram, but that's the real troiscillion in the cytoplasm. And many such troiscillions assemble on the surface of the vesicle and form the cluttering coat of the vesicle. This is the process of formation of one such cluttering coated vesicle, and you see an electron micrograph showing this process. For example, here it is the plasma membrane. That's the cytoplasmic surface, and here is the cytosol, that's the extracellular surface. We saw that cluttering coated vesicles, but from plasma membrane and also from the transgolgi network. The example here is plasma membrane. Initially, in the region where cluttering coated vesicle will be formed, in the plasma membrane there is accumulation of receptors and special adapter proteins which are binding these receptors close to each other in the region of the plasma membrane. These receptors recognize specific substance that will be attached there and transported from the extracellular space to the cytoplasm of the cell. And then on the surface of this vesicle, which starts to be formed to bud from the plasma membrane, to the adapter proteins, cluttering troiscillions become attached. And you see gradually this plasma membrane invaginates, more and more receptors, adapter proteins, and cluttering molecules become linked there, and eventually this vesicle pinches off from the plasma membrane in the form of cluttering coated vesicle. And that's the process of formation of cluttering coated vesicle. Very rapidly, when the cluttering coated vesicle is formed, the cluttering coat is disintegrated and eliminated from the surface of the vesicle. So, cluttering coat is needed for the proper assembly and budding of the vesicle, and then it is not needed anymore and it is being removed rapidly from the surface of the cell. The same happens with cautomer type 1 and type 2 coated vesicles. Okay, so the pinching off of the vesicle from the plasma membrane happens with a special cytoplasmic proteins, which are motor proteins using energy from GTP, guanosine 3-phosphate. These motor proteins are called dynamines, and they pinch off the plasma membrane there by assembling around the stem of the vesicle. And here you see many such cluttering coated vesicles being removed, or they pinch off from the plasma membrane with the help of this protein dynamine. To ensure the orderly flow of vesicles in the cell, these vesicles must be accurately recognized and directed towards their specific target membranes with which they will fuse to deliver what they transport. This specific guidance of the vesicle and recognition and interaction between the vesicle and the target structure is achieved with the help of special surface markers, receptors present both in the membrane of the vesicle and in the target membrane of the target structure. In the vesicles, we have and in the target membranes, we have the so-called RAP proteins and RAP effectors. These are directing the vesicles to specific spots on the correct target membrane and allow for the vesicle to link to this membrane. And then snare proteins and snare regulators mediate the fusion of the vesicles with the target membrane and the delivery of their content to the target structures. There are many RAP proteins described in different vesicles and different structures. These RAP proteins are monomeric guanosine 3-phosphatases, so they utilize GTP as source of energy. And today we know more than 60 such proteins located everywhere in different structures, and we can use them as some markers for these structures. We don't have to memorize them, just to realize how exact and accurate systems has the cell developed to aid the process of transportation in the cytoplasm. And these are different RAP proteins present in different membranes of target structures. And here is the interaction between the vesicle membrane and the target membrane, and how the vesicle is initially recognized by the RAP effector and the RAP GTP present in its membrane, and how this vesicle becomes attracted and binds to the surface of the target membrane. And then the snare proteins and the snare regulators interact with each other to allow fusion of the membranes and delivery of what is being transported with the vesicle. And this is a very intimate and very strict process of transportation, and it is highly regulated process. And to finish with the vesicles and the topic of our first lecture, a little bit about the special clathrin-coated vesicles, which are budding from the trans-Golgi network in specific types of cells. These clathrin-coated vesicles are called secretory vesicles. They mediate the transport of cargoes like hormones, neurotransmitters, digestive enzymes to specific sites at the cell membrane where the vesicle docks following this route that I described and fuses to release its content in the extracellular matrix. So secretory vesicles, by definition, are clathrin-coated vesicles. You see them here on this electron micrograph, black, dense, large secretory vesicles produced by very well developed Golgi complex in this secretory cell. That's the nucleus of the cell, and around the nucleus, well developed rough endoplasmic reticulum. These vesicles bud from the Golgi network, trans-Golgi network, in the form of clathrin-coated vesicles. They rapidly lose their clathrin coat and travel towards the apical plasma membrane, where they fuse and deliver their content out of the cell. Secretory vesicles are not typical for every cell. They can be seen only in secretory cells, like the cells of glands or other secretory cells. Usually they are large and can be seen even at the level of the bright field microscope, up to 1 micron in diameter. And most importantly, what you must remember, secretory vesicles are clathrin-coated vesicles. So, This was about Golgi complex and endosomes and clathrin-coated vesicles. Before we start with the topics of our second lecture today, lysosomes and ribosomes, I suggest to make the 30 minutes break a little bit earlier today. So let's make the rest of 30 minutes, and 9:30, 9:40, I'm sorry, the second lecture will start.